food allergy prevention may begin earlier than previously thought. Emerging research suggests the first months of life represent a critical developmental window when microbial exposure, immune signaling, and dietary antigens collectively shape long-term tolerance.
If a child is either, let's say, sensitized to a food allergen or is even theoretically allergic to a food allergen, so they've had not only an actual clinical reaction of some type, they also have a secondary test that's positive, skin test, blood test. What we know is that in these children, if we can actually get small amounts of the food into their diet without actually stimulating their immune allergic response, we actually can sometimes reverse this process. To me, I think that the data suggests that it's got to be under 36 months of age. I like to think that 18 months of age is probably the right number. And this is why for parents and families where there's been some type of reaction to foods, it's so important to get those kids in to see an allergist right away. We've seen some data from the group in Toronto, Julia Upton's group, which has shown that even if you can get like a 32nd of a teaspoon, literally 30 milligrams of peanut protein into kids' diets, that oral exposure really does help promote tolerance. And it seems to be that everything works better the younger the child is.
Here, we will take a look at 3 recent studies addressing the early critical window in food allergy prevention. The first study is Activate, a randomized placebo-controlled double-blind trial assessing whether exposure to maternal vaginal microbiota at birth can influence food allergen sensitization in cesarean-delivered infants at high risk for allergic disease. The findings were presented at the 2026 American Academy of Allergy, Asthma, and Immunology annual meeting in Philadelphia.
When you're born, you're essentially born with no microbes, right? And so we think that this first exposure is important because it sets the baby on the correct developmental pathway.
The Activate trial enrolled 39 vaginally delivered infants, 37 cesarean-delivered infants who received placebo seeding, and 37 cesarean-delivered infants who underwent vaginal seeding, along with their mothers. The intervention partially shifted early microbiome composition, with differences in food sensitization observed during infancy. At 3 months, the stool microbiome of infants who underwent vaginal seeding more closely resembled that of vaginally delivered infants than infants who underwent placebo seeding, though the effect was partial and diminished over time.
And it's important to remember that the Activate trial is a pilot trial. So one of the ideas behind the trial is to determine whether whether the procedure is safe, and that is the case, the procedure is not associated with increased adverse events. 2, whether we see a measurable benefit. And that's what we have shown, that there is a measurable benefit, but it's not quite yet ready to be developed in the clinic.
The second study examined whether specific diseases could be prevented by having a certain microbiota early in life. The study tracked infants born between 2003 and 2005 in Sweden, with serial fecal sampling at 8 time points from birth through 5 years of age, alongside maternal samples, blood draws, and clinical data. The team focused on a subset of bifidobacteria capable of converting aromatic amino acids into aromatic lactates. The team found that aromatic lactate-producing bifidobacteria was associated with reduced food allergen-specific immunoglobulin E. The metabolite 4-hydroxyphenolactate selectively inhibited IgE production in immune cell models.
So what we've been doing is we identified that they are there, that they are important, or seem to be important in early life, so between birth and 6 months of age. So if you have a high abundance of these aromatic lactate producing bifidobacteria, then they normally also tend to have high production of the aromatic lactates. And if you see this high production, then you see reduced development of food allergies, or at least allergen-specific IgE, so food allergen-specific IgE, as a proxy for food allergy, and also atopic dermatitis at 2 years of age. And we even also have data followed up to 14 years of age in these kids that we didn't publish yet, but seeing that hay fever and asthmatic medication was also lower in those kids with the highest abundance of these bacteria, and also the highest levels of these aromatic lactic acids.
The third study sought to identify proteins that tell the gut immune cells when to tolerate certain foods. The research helps explain why the body deems some foods as safe and others unsafe. Rather than responding broadly to all foods, the immune system appears to focus on a small set of dietary proteins that help establish tolerance, suggesting early exposures may shape how foods are interpreted. The analysis revealed 3 key epitopes derived from seed storage proteins in corn, wheat, and soybean. These findings suggest early microbial exposure may set immune development, microbial metabolites may modulate signaling, and dietary antigens may establish tolerance during infancy.
So my study looks at oral tolerance, which is the body's natural non-inflammatory response to foods, and is incredible because it is our response to most foods, right? Most people don't have allergy to most of the foods that they eat. But while we understand more and more about food allergies, we actually know comparably little about oral tolerance. What is it? How does it develop? And so I think my study is starting to identify what are the protein antigens that are recognized. And if we understand how those antigens are normally seen through the tolerance response, we might be able to apply those same principles to allergy cases to help better prevent food allergies. So we found that our corn responsive T cell population develops at the time of weaning, and then they become regulatory T cells, which tracks with what we know now in human populations where early consumption tends to lead to a tolerance response. This is why clinical guidelines now recommend if you have a baby, you should be feeding them food allergens, including peanut, when they're 4 to 6 months old. Whereas historically, guidelines were saying you should wait until they're 3. That's the wrong advice. Earlier is more likely to lead to immune tolerance. And some of my colleagues are working on figuring out what special cell types might be present at higher levels in earlier life that help to instruct that immune tolerance over an allergy response.
I'm going to steal it from the Australians. It's eat early and eat often, right? So what we want parents to do is think about their diets and think about, with considerations for, we don't want children to be exposed to foods that might be choking hazards, but really to otherwise go ahead and try and expose them to everything they're eating. I have a lot of families where they are nervous for whatever reason because maybe another child has a food allergy or the child has eczema. Those parents need to be offered an evaluation by an allergist because they may not be willing to do it at home to introduce foods early. Having said that, for all other babies, even babies with mild eczema, parents need to think about just trying to get the food in the diet, trying to minimize the skin contact, so putting the food directly into the baby's mouth and actually then exposing them to it on a semi-regular basis. That's what's been shown to reduce your food allergy risk.
It's really those first few months when it's critical to intervene if we want to lower risk. So I think a harder problem that comes out of it is, well, what happens if you have a child that already has an allergy, right? So the way I would see it is that perhaps that's a much harder problem to tackle, because we know that it's during these first few months when we can modulate the microbiome and modulate immune development based on those interventions.
These mechanistic insights may inform future prevention strategies, including microbiome-targeted interventions, metabolite supplementation, and antigen-specific tolerance. As research evolves, food allergy prevention may shift toward biologically shaping immune development during the earliest months of life.
There's 3 main factors that modulate what microbes we end up being colonized with in our life. The first one is how you're born. The second is how do you eat, broadly speaking. And the third one is, whether you're exposed to antibiotics or not, right? And I think all those 3 factors matter. So you want to be born in a way such that you acquire the right microbes to start life. Then you want to feed those microbes with the right food, and that means we need to encourage policies for breastfeeding, which is, I think, crucial. You need to introduce solid foods at the right time, right? And so, for example, with peanut, we now know that early introduction seems to have a protective effect. And then you also need to understand that the use of antibiotics needs to be well regulated. We need to not abuse antibiotics, right? So I think if we combine all those 3 factors, then you're going to see the best outcomes possible.